This is part 3 of the Chapter 12 lecture notes. And where we left off, we were talking about what a reciprocal cross is. So if it were a reciprocal cross, if the sperm gave a big P allele for purple, and the egg gave, in fact, let's do a little drawing. Sperm gave the big P allele, and then the egg, which would be bigger, gave a little P allele, and I'm making it cursive so you can tell the difference. And that's not Gregor Mendel's notation. I've learned to do that with letters that look alike when I'm doing genetics problems. So I'll just go ahead and tell you if it's an S, I'll make the big S look normal, and the little s, I'll do it as a cursive S. Um, Big C and little c are the same way. I do the little c a little bit different so that when I am doing a Punnett square problem or solving a genetics problem, you don't confuse the big letter with the little letter. So what we're seeing here is that the egg carries the little p and the sperm carries the big P. So if it were a reciprocal cross, the sperm would carry a little p and the egg would carry a big P. And then the wild type allele is the most common allele in a population. So it is the normal allele, the most common allele in a population. And the mutant allele is a rare allele in a population. Okay, and we will come back, by the way. We'll talk about wild type and mutant alleles, but not so much with Gregor Mendel's crosses. So now we're going to look at Gregor Mendel's crosses using the alleles big P and little p. So the parent generation, the purple flowered parent plant that had the genotype big P, big P. In other words, pure breeding or true breeding for purple means that the genotype is big P, or both alleles are the purple allele. And so the purple parent is a homozygote, and the white parent is also a homozygote, but a recessive homozygote, two little p's for white. And therefore, you can see if each parent only has one letter to give, then the offspring is going to have one of each. So the purple parent can only give a big P. The white parent can only give a little p. The F1 offspring don't have any choice but to have the genotype big P, little p. They're all heterozygotes. And when you cross two heterozygotes, that is called a monohybrid cross. And you're going to get these possibilities down here in the F2 generation. You're going to get 1/4, the predicted anyway, is 1/4 of the offspring are going to have two big P's and they'll show the purple flower color. Half of the offspring, 2/4 or half of the offspring, are going to show big P little, they're going to have the genotype big P little p and show the purple flower color. And then 1/4 of the offspring are going to have little p and show the white flower color. And we know that because we can do something called a Punnett square to show that that is what the outcome is. Um, So this is the Punnett square that shows the cross between the two true breeding parents. And you put the parents' gametes, the alleles that are in the parents' gametes, you put those on the outsides of the boxes. So we're putting the purple flower, two big P's on the top, and then the two little P's on the side. And then you just bring them together, and every one of the offspring has no choice but to be big P, little p. Now, we could do that in our head. We don't need a Punnett square to do that. But Punnett squares are really helpful in a situation where it's not so easy to see in your head what the combinations or the possibilities of the alleles from the parents are going to be. So one possibility when you have both parents are F1s, so both parents have a big P and a little p. One possibility is each parent will give the offspring a big P. And another possibility is that each parent will give, or that you would get one of each, a big P from one parent and a little p from the other parent. And you could get that in two different ways. And then the last possibility is that you could get two little p's, one from each parent. So that is how you would actually show these combinations here. These are the different combinations you can get from if both parents are hybrids, if both parents are heterozygous for the trait of flower color. Okay, so Mendel's first law. This is what he discovered from, this is the very first law of heredity that he generated from his garden pea studies. Heredity is particulate. That means there is a particle responsible for each trait. Each true breeding parent carries two particles, two hereditary particles for each trait. If it's a true breeding parent, that means that true breeding parent carries two same alleles. That means the true breeding parent is, the word we've learned is homozygous. The true breeding parent is homozygous for the trait. And the particles, which we now call alleles, come apart during gamete formation, such that each parent gives only one allele or one particle, or the other of a pair into each of their gametes. So if a parent has a big P and a little p, then in the formation of egg or sperm, let's draw sperm because it's easier to see. The sperm can either carry the big P or the sperm can carry the little p. And same thing with the eggs. And that way, the child is either going to get a big P or a little p from that, in this case, that father. And then the hereditary particles are recombined randomly during fertilization to produce offspring. And that is the reason we can look and get all four of these possibilities here. Okay. To test this hypothesis, he reasoned that each F1 monohybrid should have one of each of both types of hereditary particle, and he devised a formal test cross to prove it. It also became a useful means of detecting unknown heterozygotes. So a test cross, a formal test cross, really can only be done in very simple animals, but mostly you would just do a test cross with plants. But a formal test cross would be where you have a dominant individual, an individual with a dominant phenotype. So you've got this purple flowered plant, and you want to know if it's a true breeding purple flowered plant or if it is a hybrid. In other words, it has a big P and a little p. You want to know if it's got two big P's or if it's a big P, little p. So what you do is you cross it with a white flowered plant. Now, if you cross it with a white flowered plant, then normally I just see this done once, but okay, so you cross it with a white flowered plant, and then in the F1 offspring, I'm sorry, this is not really, this is showing what we already did. I'm so sorry. Okay, so let me just explain a test cross. They really haven't shown a test cross here. But what a test cross is you cross the purple that you don't know its genotype with a white that you don't know its genotype. Forget all this stuff over here. Um, and if you get any offspring that have the white color, then you know that this parent is big P, little p, a carrier of the white allele. Sorry. And if you don't, if all the offspring are purple, you can be pretty sure that the parent has two big P's. So that's what a test cross shows. And then down at the bottom it says predict a 1-to-1 ratio of dominant to recessive offspring in the F2 and why. Um, so that is because we know this F1 offspring is big P little p, and this is little p. So if we do a Punnett square, see, I think I've got room, where we cross the big P little p with the two little p's, we get big P, big P, little p, little p, little p, little p, little p. So that is 50% white. And that's where that 1-to-1 ratio comes from. A 1-to-1 ratio of dominant purple color to one white recessive white color. Here it's showing 4-to-4, but that's the same thing as 1-to-1. Okay, and I've shown you the Punnett squares already, so let's come back and do another test cross because this one is easier. Um, in a test cross where we're looking at seed color, big Y is the allele for yellow, which is actually the dominant allele, and little y is the allele for green, which is the recessive allele. And we have an unknown dominant yellow seed plant that we want to know if it's big Y or big Y little y. Okay? So when we cross that yellow seed with a green seed recessive plant, if we get any green offspring at all, then that parent, that unknown parent, must have been big Y little y. If all we get are yellow offspring, then that unknown parent is big Y. Okay? And then it says Punnett squares can predict Mendelian cross outcomes. Each homozygous parent in the P generation produces only one kind of gamete, either a big Y or a little y, because that's all the yellow parent has to give is a big Y, and all the green parent has to give is a little y. The F1s produce two kinds of gametes, a big Y and a little y. Even though they're all yellow color, their phenotype is yellow, their genotype is heterozygous. So they're going to produce two kinds of gametes. And then the F2 are going to produce three total different types of gametes. They're going to produce either two big Ys, or they're going to produce a big Y and a little y, and that can happen two different ways. Either the sperm gives the big Y or the sperm gives the little y, and the egg gives the other one. And then the other possibility is two little y's. So when we look at the genotypes, the ratios of each genotype, it's 1 to 2 to 1, 1 big Y, 2 big Y, and 1 little y. The phenotype ratio is 3 yellow to 1 green. And that does not mean that that is just the predicted ratio. So parents, these two parents, the F1 parents, the parents are big Y, little y. They're big Y, little y crossed with big Y, little y. The predicted outcome is that they'll have 3/4 of their offspring that'll be yellow and 1/4 that'll be green. But it could happen that all the offspring are yellow. It could happen that all the offspring are green. It's not very likely. What's likely is that 75% will be yellow and 25% will be green. But remember that these are predictions. And the more offspring that are generated, the more likely that they will follow the predicted results. A little bit of information about probability. If they're chance events, they're described by probability. And when we do Punnett squares, that's what we're doing, is we are looking at probability.